today is binding of hepatitis E virus RNA-dependent RNA polymerase to the cis-acting regulatory elements in the HIV genome. Basically RNA-dependent RNA polymerase is an enzyme which catalyzes the synthesis of RNA strand from the RNA template. And this RDRP is a key enzyme which regulates the basic virus biology in RNA viruses life cycle. So, initially some key facts about the hepatitis E virus. Hepatitis E is a liver disease caused by the hepatitis E virus. It is a non-enveloped positive sense single-stranded RNA virus, and basically the mode of transmission is a fecal-oral route, and it is basically transmitted by contaminated food and water. And although it is a acute and self-limiting disease, it can cause sporadic fulminant hepatic failure in adult and children also. And according to WHO, almost 56,000 cases per year was reported from all the 20 million hepatitis infection cases, over 3 million symptomatic cases. And although it is acute and self-limiting disease, it can cause a high mortality rate in pregnant women, and the reason for it is still not clearly understood. And chronic cases in immunocompromised and organ transplant patients are also reported. And in India, 30 to 60% of all sporadic cases of hepatitis are due to HIV infection. And it can also be a zoonosis. That means it can transfer to animal to human, and that is the major concern, and you need to understand the virus basic biology. And currently no antiviral drug is there for HIV, but recently one vaccine has developed by China, but it is not available rest of the part in the world. And this is the electron micrograph of hepatitis E virus. It is basically 32 to 34 nanometers spherical non-enveloped particle with surface spikes, and it belongs to a single membrane as family Hepaviridae in genus Hepavirus. Basically it has 4 genotypes, genotype 1, 2 and 3 and 4, and this is the geographical distribution of HIV. Here the almost all Asia, Africa and Central America is the highly endemic area for hepatitis E. And since we don't have any cell culture system or small animal model for hepatitis E virus, so we don't know the basic virus biology about it. So based on the domain homology between hepatitis E and other positive RNA strand viruses, our replication model has been proposed for hepatitis E. So this is the replication model, and once the RNA genome gets entered into the host cell, it acts as a mRNA for mRNA inside the host cell, and it hijacks the host translational machinery, and it translates its non-structural protein including viral RDRP. This viral RDRP further binds to this positive strand RNA, and it replicates into the negative strand full length RNA, as well as a small subgenomic positive strand RNA to express its downstream proteins, which further produce the structural protein and other protein which is required for full virion production. And this full length genomic RNA and this capsid protein is assembled to make the progeny virions. And here the mechanism about the subgenomic mRNA synthesis is not understood. We don't know that this subgenomic RNA is produced from full-length negative sense genomic RNA, or it produced from the small-length negative sense RNA, and then it make small mRNA for structural protein production. And the subgenomic promoter region where the RDRP binds and produce subgenomic RNA is still not fully characterized or not identified yet. It has just proposed the region, but nobody has characterized it. And these regulatory elements by which the transcription, translation, and replication was controlled in the genome is not still identified. And mechanism of viral RDRP by which it produces full-length viral genome or subgenomic RNAs are still has to be explored. And this is the previous study which has proposed the cis-regulatory elements on viral genome. It has kept 5' UTR 26 base hairpin at its 5' end, and it can act as a genomic promoter in the negative sense RNA for the production of full-length genomic positive sense RNA. And this is the 5' end. This in the ORF1 region, a 58 nucleotide conserved sequence is also found, which is homologous to alpha viruses, and it forms 2 hairpin. It can also act as a regulatory elements to bind with the RDRP. And this in ORF1 region, small region is found which is homologous to SYNBIS-like junction site, which is the promoter region for the SYNBIS virus. So it can also be act as a promoter for the subgenomic RNA synthesis. And another region has found between the ORF1 and ORF2 junction region, and it has a stem loop structure, and it is reported to important for the virus replication. And finally, the 3' UTR region, it is now has been characterized as a RDRP binding site in hepatitis E virus genome to produce the full-length negative sense RNA genome. So in this our study, we have made different constructs using these areas. Basically, we designed primer against the this areas, and it has a T7 promoter in 5' or 3' end to produce positive and negative sense RNA template to test the RDRP binding efficiency. And this we PCR amplified these templates, different templates, and then we in vitro transcribed to produce the its corresponding RNA. And these are the tabular form which represent the different fragments we tested for the RDRP binding. And first we cloned the RDRP PCR amplified RDRP in PET15B vector, which has N-terminal histidine tag, which is used for further purification and detection of RDRP. And 2 rounds of purification was done. First it was used as in affinity chromatography by using probond nickel chelating resin column, and then second round was done as gel filtration chromatography by HPLC. And simultaneously we made active site mutant of RDRP, that is GDD to GA mutant, by site directed mutagenesis. And this is the purified RDRP protein which is further identified by Western blotting, and this is affinity purified fractions detected by Western blotting, and this is the second round of purification by HPLC. Similarly, it was done for GDD mutant for RDRP protein. And further we developed the electrophoretic mobility shift assay for using 3' positive sense NCR as a template, because it is already reported that RDRP binds with the this 3' NCR of the positive sense genome. So this is the methodology. I'm not going to explain this. This was the EMSA done by the different positive and negative sense strand of generated strand, and this positive strand EMSA shows that subgenomic 5096 positive strand and 3' positive strand only binds with the RDRP. Rest of the fragments doesn't show any binding. Similarly, in negative sense strand, there is a specific binding against the subgenomic 5096 negative sense template. And similarly, the 5' extended NCR region, which included the 58 nucleotide conserved region, also shows the binding with RDRP with both the positive and negative sense RNA. And similarly, we have done the template competition assay to identify the specific binding and affinity for the RDRP. So we did a competition assay. The first A and B is the assay for the subgenomic 5096 positive sense, and this code, this is non-radiolabeled subgenomic negative sense was supplemented with this strand in equimolar 50, 100 and 200 fold axis here. And it shows that both when we did the positive sense and negative sense competition assay, both has similar affinity with RDRP, and it has inhibited the binding of other strand. And similarly, when we did a 3' NCR template competition with the cold subgenomic negative sense template, it has observed that subgenomic strand has partially inhibited the binding of RDRP in 3' NCR template, whereas the excess amount of 50, 100 and 200 fold excess amount of template shows the super shift kind of observation, and it might be the co-binding of both the fragment with the RDRP. And similarly, when we did it with subgenomic negative sense was supplemented with 3' positive sense cold RNA in similar fashion, and it shows the same results as it was in with the competition with subgenomic and 3' NCR region. And both the results shows that there was super shift, and it was it was a co-binding with either these two RNA independently or in presence of RDRP, or either RDRP has two binding site for both the RNA. Yeah, and again we tested this 5' NCR region, which is extended with 58 nucleotide conserved region, and both positive and negative sense was competed with each other. And here this when we competed negative sense with positive sense, the binding was inhibited. And similarly, the 3' NCR in equimolar amount and 20-fold axis was also inhibited the binding of this 5' NCR to 50 positive sense binding with RDRP. But this subgenomic 5096 fragment was showing a partial inhibition in almost 24 axis. So, and similarly, when subgenomic negative sense was competed with positive sense of its fragment and 3' positive sense and subgenomic negative sense at equimolar and 20-fold axis, it shows that positive sense doesn't inhibit the this negative sense binding of RDRP. Similarly, here the 3' NCR was completely inhibit the RDRP, where a subgenomic fragment was showing partial inhibition with the binding with RDRP. So binding affinity was observed. Finally, was the 3' positive sense was the highest the highest affinity with the binding of RDRP, then 5' NCR, and then subgenomic negative sense this template. with RDRP. And then we performed the experiment to identify if there is two binding motif in RDRP for genomic and subgenomic RNA template. So this is the methodology, and we performed the protease digestion assay. We first performed the EMSA for both 3' positive sense and subgenomic negative sense RNA, and then we sent that sample for mass spectroscopy, and terminal sequencing and QTOF was done, and it was analyzed, and this program were used for 3D structural modeling and prediction of the RDRP structure with RDRP, and we found that there is two peptide was found for subgenomic 5096 negative sense strand, and there was 3 peptides were identified for 3' positive strand template, and here the 2 peptide was similar in both the template, but here the one single peptide is extra for 3' positive sense RNA, and it can be the region that 3' positive sense has a higher affinity for RDRP. And this is the conclusion. The competition assay suggested that HEV RDRP prefer binding to 3' positive sense when there is low concentration of subgenomic negative sense, meaning that synthesize more negative sense RNA once there is accumulation of higher concentration of the negative sense molecules. It binds to subgenomic negative sense promoter region, probably via other motif on RDRP, while the enzyme is still bound to 3' positive sense. The competition of 5' NCR 250 PS with negative sense revealed that at the equimolar and 20-fold excess concentration of the cold subgenomic negative sense, 5' NCR negative sense, there was significant inhibition of the binding of positive sense with HEV RDRP. Considering that 3' end of the replicative intermediate negative sense RNA would be putative promoter of the RDRP to initiate complementary strand synthesis. And here the peptide was identified for the binding of this subgenomic and 3' template, and that could be the third peptide may help in higher binding affinity for RDRP for 3' NCR. That's it.
Thank you, Shakuntala. I suggest that we skip the questions, and if anyone has questions, we can address them later in the break, because we still have one talk. I think there is Soza Babam, right? Okay. So
Okay.
thank you very much, and just please take any questions in the break.
Okay.
Thank you very much.
Thank you.
Yeah.